For the remainder of this week and into Wednesday of the week when we return from spring break, we're going to cover all aspects of neuronal plasticity, learning, how the brain rewires itself, as well as at the end, repair of damaged neurons. So we're going to talk about like neuronal repair and those mechanisms that are involved in that as well. But for now, let's go ahead and first talk about synaptic plasticity. So these are the things that we're going to cover in today's lecture. We're going to talk about short-term synaptic plasticity, which involves these four major mechanisms: synaptic facilitation, depression, as well as post-tetanic potentiation. We're mostly going to refer to it as potentiation instead of PTP. And then there's augmentation as well, which is very similar to post-tetanic potentiation, except it's a little bit shorter lived. But they're kind of like different sides of the same coin, basically. We're going to discuss habituation versus sensitization. We're going to also talk about long-term potentiation and long-term depression. And then we're going to discuss how glutamate can play a role in these different types of neuroplastic effects. And then we're going to talk about different types of seizures, which could involve excessive excitability of the brain. Right? And there's going to be different seizures that I'm going to have you guys responsible for. So that's going to be the more fun aspect of this lecture, the clinical manifestations of everything that's going on here. So this is short-term synaptic plasticity, and these are the major things involved in that. So you have synaptic facilitation, and that's going to involve increasing neurotransmitters, which allow for, you have increased neurotransmitter release as a result of calcium. And then you're going to get more temporal, which means over time, summation of activity in those neurons that are affected. Synaptic depression is going to be the opposite, where you have neurotransmitter release, but then you get a decline, right? And the decline is going to occur during the sustained synaptic activity. And so it's going to involve different types of pathways. And then you have post-tetanic potentiation, where you have excessive stimulation of the neuron. You get a bunch of calcium entering into the neuron. Then you get increased neurotransmitter release. And this one's going to last longer than augmentation. This is going to last like seconds and then into minutes as well. It can last several minutes. And then augmentation is basically potentiation, but it's short-lived. It's only going to last a few seconds.
Basically, PTP and augmentation are versions of synaptic facilitation.
Sort of, not really. There are different processes. There's a graph from your textbook that actually shows the differences between these and how they kind of interplay with each other. But no, these are all different mechanisms. So they're not variations of each other. They're all different mechanisms, except for augmentation is kind of a variation of post-tetanic potentiation, just shorter lived. So here is a study that they were using giant squid synapses. So you see facilitation. So whenever you see facilitation, that basically augments the excitatory postsynaptic potential, right? So when you have facilitation, it makes those responses a little bit more intense. They augment it. They increase the amplitude of the depolarization. And facilitation will, over time, decay. So you can get this is time dependent. So facilitation, for example, over here is going to be greatest at around 0.8 or so milliseconds. As the milliseconds continue on, like around 50 milliseconds, that's when facilitation drops tremendously. So facilitation is time dependent. So here's another image that talks about, how you can, graphically represent short-term synaptic plasticity. So they're giving a stimulus over here, and it's going to be like a high tetanus type of stimulus. So everything that's here in the shade is going to be kind of a tetanic type of stimulation of the neurons. Then calcium is going to be involved in this process. So if you have normal levels of calcium, you're going to have a normal sustained effect. If you have intermediate levels of calcium, it's going to start resulting in more like augmentation. And if you have a very low amount of calcium, the effects are going to be a little bit reduced as well. So that's all your tetanus type effect, so tetanic effect. So here's another image of that. So you have your stimulation, which is inducing a tetanic effect, and then this is post-tetanic potentiation. And post-tetanic potentiation, or PTP, is going to last, again, like seconds to minutes. So this can last for a while, a little bit longer than augmentation. So you have, again, various forms of these short-lived type of plasticity events. Calcium is going to be involved in all these different effects. You can have facilitation and augmentation being involved in these processes as well. So here's that image I was telling you about that gives you a good depiction of what's going on here. So you have facilitation, then you have augmentation, then you also have depression, and then you have potentiation as well. So there's a bunch of different mechanisms that come to play, and they all involve different types of neurotransmitters. So we're not going to go into specific neurotransmitters here, but we already learned a few of these. It could be like glutamate, it could be GABA, it could be a bunch of different neurotransmitters that are involved in these different processes. So this first line over here, this is where they have the actual stimulation, right? So presynaptic stimulation of the synapse right before, right, the postsynaptic cell. So you have tons of like repeated stimulating signals being sent to that neuron. At some point, you start seeing the effect of the stimulation decline, right? So you have increased postsynaptic stimulation from the presynaptic cell. So you have increased amounts of reaction until at some point it peaks out. So it peaks out and it can't go on any longer. That's when you start getting things like augmentation coming into play. And like I said, with these different processes, they're going to involve different types of neurotransmitters. So you might have too much stimulation and all of a sudden you need to bring things back down again. Neurotransmitters are going to come in to actually help to decrease the amount of stimulation. So that would be the form of augmentation. Once you stop stimulating that neuron, everything basically goes back to normal. However, you're going to see augmentation and facilitation kind of decay over time. Then if you wait a little bit longer to stimulate that same neuron again, you might see post-tetanic potentiation involved in that process. So these are all different examples of like short-term plasticity. So again, these are like the types of they're dynamic changes. They're going to be involving neurotransmitter release, and these are just different forms of short-term potentiation, or sorry, short-term plasticity. Short-term mechanisms of plasticity cannot account for the long-lasting changes that hallmark learning and memory. So these are just going to be short-term type changes that affect the cells. But there's going to be long-term changes that are going to be more associated with learning, memory, things that are going to take place, for example, within the hippocampus, right? Whenever you're learning a new skill or learning, if you're encoding memory for long-term retrieval and storage, there's different mechanisms that come into play there. So long-term synaptic plasticity. Some of the original studies in this involve the giant sea slug. So that's the Aplysia. And Kendall, he's the researcher that did these studies decades ago. He actually received a Nobel Prize for all of his work on habituation as well as sensitization. So when you habituate, what we mean by habituation is that if you repeat a stimulus to a person or an animal, over time, that stimulus is not going to elicit the same type of response, right? So we actually do this in a clinical setting when we're doing a test on a patient, and we could even do this to each other. It's called the glabellar tap sign. So what you do is you have the patient have their eyes open, and then you literally tap their glabella. The glabella is the space between their eyebrows. And the first response should elicit like a blink, right? That's kind of like this the natural response and a natural reflex to the glabella tap. It's like you're wincing, basically. Or what's the term for that? When someone like flinches? Yeah, you flinch, basically. And then when you keep on tapping with repeated taps, you should basically habituate to it. And you no longer flinch, and you no longer blink your eye. But there are some people who have neurological conditions where every single tap, they're still flinching, they're still blinking their eye, right? That means that their capability of habituating to the response or the stimulus is no longer intact. It's not working properly. So let's watch a little video on this. So this is a patient who's not able to do a proper reflex.
This sign is usually positive in Parkinson's disease. When in a normal person, if we tap the glabella, then for the subsequent or initially when we start tapping, after 2 to 3 taps, for the first 2 to 3 taps, the eye will blink, but after that, the subsequent taps, eyes won't blink. But in the case of Parkinson's disease, when we go on tapping, with each tap, the eyes will continue to blink for a prolonged period of time, irrespective of your number of taps. This is known as glabellar tap sign, which is seen in Parkinson's disease. Now, in this patient, let us see what is glabellar tap sign.
So you see he's constantly blinking his eyes. Let's do that to each other. Let's do it. Get a partner and tap on each other's glabella. Do it for like the next 30 seconds. I'm serious. Partner up. Do it. Yeah. See how many times you can. See how many times it takes for the habituation to take place. If you want, there's a sanitizer over here. If you feel grossed out and you don't want to get each other's cooties, but tap on each other's glabella. And then on the weekend, when you're hanging out with your friends, do the same thing. Tap on each other's foreheads while you're drinking. See if any of never mind. I take that back. Yeah, literally, you just tap on the glabella. See how many times it takes for you to stop blinking. And just like, tap, 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 tap.
You didn't blink at all the whole time. Maybe because you knew it was coming.
I was like, I don't have Parkinson's.
You don't have Parkinson's? That's good. I'm glad to hear that. It should stop at some point, right? That's called habituation, right?
But then as soon as I stopped for a second,
So how many times did it take for you to tap for the stop? It was one. So after like 1 or 2 taps, you stopped blinking, basically. You didn't blink at all. That's really weird. Just kidding. You have a condition. We were going to have to diagnose you with something. We have to diagnose you with something, at least. So that's habituation, right? So you habituate to a stimulus in the environment. Now, sensitization would be the opposite. Sensitization, you're going to increase the amount of reaction you have to a stimulus. So the type of work that was done to be able to figure out what's going on neurologically with these mechanisms involved this giant sea slug. So it's a super cool animal. So let's play a little video here.
This is a good model to study learning and memory. I don't know about you, but I feel sluggish sometimes, especially before I have my coffee. But who knew these sea slugs would have so much to tell us about the human brain? Our brain and nervous system are wired with hundreds of billions of nerve cells, or neurons. But slugs keep things simple, with just tens of thousands.
When we talk about learning in a slug, we're talking about how to find what to eat, how to avoid being eaten, and how to find a mate and reproduce. Simple behaviors. So you can actually study learning and memory and how neurons control behavior probably better than any other creature.
With support from the National Science Foundation, analytical chemist Jonathan Sweetler and his team at the University of Illinois are working to understand the function of individual cells in the nervous systems of slugs and other animals.
So why study single cell? Trying to understand how, say, a muscle works, and maybe individual cells aren't important, but in cases of cancer, when the single cell starts to mutate, then you need to go to the single cell level.
They use a technique called mass spectrometry to measure the different chemical constituents in slug and other brain samples.
Every one of these lines is a different chemical from the brain. So because of what's happened with mass spectrometry, the discovery of new molecules in the brain is really accelerating.
Sweetler says sea slugs are a great model system for neuroscience because their neurons grow and grow throughout their lives.
This is the nervous system of the aplysia californica that we were looking at. And right here, you can see that some of these neurons can actually get quite large, which makes it a little bit easier. There's a little more material to work with.
Learning to turn specific chemicals in the human brain on and off could lead to new methods to diagnose and treat chronic pain, drug addiction, even neurological disease and mental illness.
And so we want to understand the chemistry. And more than that, we are discovering in every animal we've looked at new molecules that people didn't know were in the brain. Because as we find new molecules, there is actually the potential for having a therapeutic effect.
So next time you're feeling a little sluggish, take heart. They're smart enough to teach us a few lessons. For Science Nation, I'm Miles O'Brien.
I'm going to show you another video in a second that shows the habituation and sensitization of those slugs. Not a super great video. There's actually not that many videos out there that show the withdrawal reflex, but this was the work of Eric Kendall. And like I said, Eric Kendall's work awarded him the Nobel Prize. Actually, I think this is the video on him. Yeah, let's watch this video. This is where he's talking about what's going on with his research.
Very satisfying way to determine, pick a problem that is really important, try to tackle it in a very, very serious way. Don't do a trivial problem. It's better to make small progress on something really, really important than to push a trivial issue further ahead. My scientific questions often come from things that I have personal interest in. I went to Harvard and majored in history literature because I wanted to understand what happened to me in Vienna, 1938, when Hitler marched in. I wanted to see how, civilized people could become so brutal and anti-Semitic. While I was writing my dissertation, I met a famous psychoanalyst, and he said to me, if you want to understand motivation, if you want to understand what happened to you, you're not going to do it through history and literature. You've got to really study the mind. You've got to study why people do things. You've got to study psychoanalysis. I had no science background whatsoever. I took physics, I took organic chemistry, took the courses necessary for medical school, and I went to medical school with the idea of becoming a psychoanalyst. I came along at a very good time when neuroscience and psychology, which were worlds apart, was beginning to come together. I was one of many people that contributed to that. There were people before me that already started the way. But I took advantage of the fact that this was a new direction that was opening up. And there were certain experimental systems that were particularly conducive to that. And many neuroscientists of my generation were scared to work on behavior because they thought it was too complicated. And I, being a psychiatrist, that didn't bother me in the slightest. So I learned how to study behavior, worked out a simple reflex that I could study, and see what happens when an animal learns something. And I found that what happens is that the strength of synaptic connections changes. The nerve cells communicate with each other at specific sites called synapses. And these synapses are changed by learning. And certain kinds of learning, the synapses get stronger. Other kinds of learning, they get weaker. Well, I think working out the first cellular mechanisms of learning and memory is not so bad. I'm quite proud of the fact that I did this. Way beyond that now, the field, which was very small when I came into it, is now gigantic. I mean, many people work on the brain, different aspects of behavior, and making very good progress. I mean, the brain is unbelievably complicated. We've got decades and decades of work ahead of it, but very good progress.
Yeah, really cool dude. Anyways, like I said, so he won the Nobel Prize on a lot of his work on habituation and sensitization. So here is the Aplysia giant sea slug. These are all of the neurons involved. You got different neurons, ganglia. That's where you find a lot of your cell bodies and dendrites and things like that. And these are going to be involved in the gill withdrawal effect, right? So gill withdrawal effect, which they showed a little video of that. So the first time you touch the siphon, right, it's going to contract very aggressively at first. But then you can train with repeated touches over time to basically allow for habituation, right? They get used to it, right? And then so the effect is not going to be as significant as the very first withdrawal effect, right? So that's really great example of habituation. Then if you stimulate the neuron using electric stimulation, you can actually do the opposite. Then you can increase the sensitivity to the stimulus. And so that is sensitization. And so there's different types of experiments where they did that, where they would like administer more of a depolarization effect with electrical voltage to sensitize the animal to have a more aggressive reaction. So in other words, tail shocks are going to prolong sensitization, right? So here's the video. Oh, that's not the same one. Sorry, that's a different one. There's another video that I think I put in here that shows that like siphon withdrawal effect. But anyways, the gill withdrawal response, again, for aplysia, it's going to be a very simple type of circuitry. It involves interneurons, right? And interneurons are especially inhibitory. It can also involve motor neurons. And with habituation, it's going to be moderated by the glutaminergic pathways. So it's going to be glutamine that comes into play, right? Remember, glutamine is your excitatory neurostimulator. So if you depress glutamine, then that's going to allow for habituation. So in other words, depressing glutamine is going to allow for habituation. If you overstimulate with glutamine, then that would cause sensitization. And so here's how they started to induce sensitization. They would put this little stimulus here, so they would start shocking the tail neurons, and when in so doing, they allowed for more of a sensitized response. So here, for example, these first 50 minutes where they would just repeatedly activate the snail to be able to become habituated. Over time, the response, became less and less. Right? So here's your excitatory postsynaptic potential in the motor neurons. Over time, with continued habituation, those reactions became less and less until you stimulate the tail nerves, and then you induce sensitization. And now look at the excitatory postsynaptic potential. It's even greater than it was without any stimulation. Right? So you can induce sensitization. Now, this could also involve serotonergic pathways too. So if you have serotonergic modulation of those interneurons, that could induce a more robust response with sensitization. So short-term sensitization is going to be serotonergically mediated. It involves the whole entire second messenger cascade, which I'm not going to hold you guys responsible for, but know the neurotransmitters are involved in these processes. So habituation, reduction of glutaminergic pathways, right? Reduce glutamine. Sensitization is going to involve serotonergic pathways. And you're also going to see enhancement of the glutaminergic pathways as well as a response. Now, with long-term sensitization, you're seeing more gene expression being involved. So not so much the effects of the actual neurotransmitter itself, but you're seeing more of gene transcription and translation into different proteins. So it leads to the synthesis of proteins required to modify existing synapses. And then so you get changes in the synaptic growth. So that's long-term sensitization. Now, you can interfere with that by playing around with different genes, right? So if you use Drosophila, for example, you can mutate second messenger pathways. So I'm not going to hold you responsible for these different genes, but there's like a couple of them. One, like DUNS, for example, affects phosphodiesterases. If you affect adenylate cyclase, if you affect other peptides that are involved in that process, then you might see issues with the whole entire learning and memory process. So for just these Drosophila experiments, they're looking at olfaction, the olfactory learning task. And if you mess with any of these genes, you can basically inhibit their ability to learn through the olfactory memory task. And different genes are going to have different degrees of severity of effect on learning and memory. Now, in terms of the hippocampus, right, all of this is going to be really important for learning and memory, right? And learning and memory is going to be especially involving the hippocampus in the brain, right? That's where you get long-term storage of information, for example. So long-term potentiation and long-term depression are going to affect the ability for memory to take place within the hippocampus. So with long-term potentiation, you get long-lasting synaptic strengthening. You get pattern. Geez, I messed that up. Pattern increases. I should say pattern increases of synaptic activity. So that's a typo. I need to fix that. So sorry about that. Versus long-term depression, you get decreases in synaptic strength. So in other words, you are either strengthening a synaptic bond with, for example, long-term potentiation, or you're going to weaken that synaptic bond, right? And when you create more and more synapses between two cells, that's when you get, more communication between those cells. Synaptic pruning, when you're like taking back those synapses, you're going to reduce the strength of those connections. There's a quote, neurons that fire together wire together. And if you have more neurons firing together, they're going to have an increased strength. So this is all part of neuroplasticity, right? Creating synapses, taking away synapses, right? When we say neuroplasticity, we're talking about changes in the way that the neurons are able to fire together. There's this long video here that talks about long-term potentiation, and they do a really good job at explaining it. So I do want to play this. It's about never mind. This is 2 minutes. There's another video that's a bit longer that I'm going to include in that whole hippocampus process. I think that's actually part of the next series. I forgot about that. So let's play a short video here on long-term potentiation within the hippocampus.
Welcome to 2-Minute Neuroscience, where I explain neuroscience topics in 2 minutes or less. In this installment, I will discuss long-term potentiation, or LTP. LTP is a process by which synaptic connections between neurons become stronger with frequent activation. LTP is thought to be a way in which the brain changes in response to experience, and thus may be a mechanism underlying learning and memory. There are a number of ways in which LTP can occur. The best known mechanism involves a glutamate receptor known as the NMDA receptor. In NMDA receptor-dependent LTP, glutamate release first activates a subtype of glutamate receptor known as the AMPA receptor. NMDA receptors are found nearby these AMPA receptors, but are not activated by low levels of glutamate release because the ion channel of an NMDA receptor is blocked by a magnesium ion. If frequent action potentials cause greater stimulation of AMPA receptors, however, this will cause the postsynaptic neuron to depolarize, which eventually causes the voltage-dependent magnesium blockage of the NMDA receptor to be removed, allowing calcium ions to flow in through the NMDA receptor. This influx of calcium initiates cellular mechanisms that cause more AMPA receptors to be inserted into the neuron's membrane. The new AMPA receptors are also more responsive to glutamate and allow more positively charged ions to enter the cell when activated. Now the postsynaptic cell is more sensitive to glutamate because it has more receptors to respond to it. Additionally, there are thought to be signals that travel back across the synapse to stimulate greater levels of glutamate release. All this makes the synapse stronger and more likely to be activated in the future. This process is also associated with changes in gene transcription in the neuron, which can lead to the production of new receptors or modifications to the structure of the cell. These changes seem to be important for making the increased responsiveness of LTP long-lasting.
So again, long story short, glutamate is going to be really important. Sorry about that. Glutamate is going to be a really important neurotransmitter involved in this whole process. Now, here's how the hippocampus works. You got this trisynaptic circuit. So you have different regions in this, the hippocampus that are going to communicate with each other. And this is where you have long-term potentiation. So each of these little plus signs in this image over here, that's all areas where you're having long-term potentiation. And whenever these get stimulated, that's when you start getting like memory formation, right? That's where you actually get memories being formed. Here's that long video that I wanted to play. So this one's about 4.5 minutes long. So I'll play that because it goes through and does a really good job of describing this whole, like the trifecta, right? You have like 3 different synapses being involved in this whole process. The pyramidal cells in the neurons are going to be involved in this. You also have Schaffer collaterals or Schaefer collaterals. So Schaefer collaterals kind of work in parallel, so they work in tandem. And then you have pyramidal neurons that are going to be involved in this process as well. Like the whole entire idea here, though, is to have long-lasting increases in excitatory postsynaptic potentials. Right? That's where you get long-term potentiation, and that's where you get memory formation and memory storage that lingers for a long time. So let's watch another video on this. They do a really good job of describing this whole process. So let me start from the beginning here.
This video aims to explain the structural architecture of the hippocampus and the transmission pathway of the trisynaptic circuit, all of which are associated with cognitive processes like memory formation. To start with, here is the anatomy of the brain. This is visually and structurally divided into 4 lobes: the frontal, temporal, parietal, and occipital lobe. Deep within the medial temporal lobe is a set of structures collectively known as the limbic system. We will solely be focusing on the hippocampus, which is one of the structures that make up the limbic system. This is a paired structure, with one found within each cerebral hemisphere. The word hippocampus is Greek for seahorse, as it takes on the shape. It's part of the hippocampal formation, comprising of the parahippocampal gyrus, entorhinal cortex, the subiculum, and the dentate gyrus. The hippocampus has roles within spatial awareness and the conscious recollection and consolidation of declarative memory. These are the types of memories that you can declare, like facts and events, known as episodic memory. Case studies support its role in memory formation, where lesions in the hippocampus have resulted in the inability of patients to form new memories. However, it's important to note that there are many types of memories, each supported by distinct neural systems throughout the brain. For example, non-declarative memory, which includes procedural and habitual learning, is known to be supported by the basal ganglia. Now, let's look further into the trisynaptic circuit, which will explain the flow of information through the structures of the hippocampus. The major sensory input into the hippocampal formation will come in from the entorhinal cortex. This is a bundle of fibers forming the input tract. This will act as a hub in the widespread network for the memory. These bundles of fibers in the entorhinal cortex will project to the granule cells of the dentate gyrus via the perforant pathway. This is the first pathway in the trisynaptic circuit. From here, the dentate gyrus will transmit its signals to the CA3 pyramidal neurons through the mossi fiber pathway, which is the second pathway of the circuit. These mossi fibers have dense reciprocal connections which are able to generate new patterns of activity, where one single mossi fiber can project to around 35 pyramidal cells within the CA3. CA stands for cornu aminus and helps to divide the hippocampus into 4 subfields, from CA1 to CA4. This name actually comes from the similarity between the shape of the hippocampus to the ancient Egyptian deity Amun, who has horns of a ram. Then the Schaefer collateral pathway, which is the third pathway completing the trisynaptic circuit, will connect the CA3 neurons to the CA1 Schaefer collateral neurons, where it has the highest concentration of NMDA receptors within the brain. Finally, the CA1 neurons will project to the subiculum, which is considered the major output region for the hippocampus. It will go out to the cortical and subcortical regions. The subiculum not only receives cortical inputs from the entorhinal, pararhinal, and prefrontal cortices, but it sends projections out to the fornix and back out to the entorhinal cortex in order to complete the loop. All of the cells that make up the hippocampus will collectively act as an index. This is similar to how a computer index works, as it's able to retrieve and record the information that is necessary for particular memories. In the case of playing piano or studying, the The hippocampal index will retrieve and reactivate all of the distributed parts of the different processing areas associated with that memory. So essentially, the stronger the connections are between these neurons, the quicker you'll be able to recall what you wanted to remember. This is essentially a cortical representation of a memory. Therefore, the hippocampus has to communicate with many distributed regions of the cortex and to collect all of the information from these widespread cortical areas. If you like this video, please leave a like and press that subscribe button if you want more.
So that's just an awesome video that breaks down the whole entire pathway within the hippocampus. Do I want you to know that pathway? It's a lot. No, I'm not gonna test you on all that stuff. Know some of the major neurotransmitters involved, like glutamate. Right? It's good to know those receptors. You got AMPA receptors, right? NMDA receptors are involved in that. The AMPA receptors do not have magnesium blocking the entry pathway, right? So these are ion channels, right? But like the AMPA receptor does not have magnesium, but the NMDA receptor does, right? So the NMDA receptor is going to be inactivated when it has magnesium in the middle of it, right? So it obstructs flow of calcium ions. So just know the different neurotransmitters involved, right? Glutamate, super important for memory formation. This is all FYI. I'm not going to test you on the Schaffer collaterals. I'm not going to test you on all the CA1, CA2, all that stuff. I'm not going to test you guys on that stuff. So these are all very, very convoluted pathways in memory consolidation, memory formation. So let's move on here. Here's one thing that's kind of interesting. So recording the excitatory postsynaptic potential from the hippocampus in vivo reveals that high-frequency stimulation can produce long-term potentiation that lasts more than a year, right? So these effects have long-lasting effects. Or these processes have long-lasting effects. Long-term potentiation requires strong activity in both pre- and postsynaptic cells. So we're going to talk about next time we meet, we're going to talk about the quote, synapses that fire together, wired together. Right? The more synapses are going to be coming into contact, the stronger the connection is going to be, the better ability you have for long-term potentiation. These stimuli alone do not elicit any changes in synaptic strength. However, brief polarization of the CA1 neuron's membrane potential in conjunction with the Schaffer collateral stimuli results in persistent increase in excitatory postsynaptic potentials. So that's the long story of this, right? The more you stimulate these nerves, the more they're going to be able to maintain connection. In other words, the longer the memory is going to last within these neurons. Long-term potentiation, synapse and input specific, right? So you have associativity, right? So they associate with each other. The more synapses that you have in connection, the better associations you have. Weak stimulation of one pathway will not lead to long-term potentiation. You have to have those strong connections between the two. Strong stimulation of adjacent pathways will increase long-term potentiation. So again, neurons that fire together, wire together. So here is example of specificity, and then you also have associativity. So with specificity, you have, the more important thing here is the associativity. When the same weak stimulus to pathway 2 is activated together with strong stimulation to pattern 1, both sets of synapses are strengthened. So if you have more synapses doing a similar type of signal, even if it's a weak signal, just the presence of that weak signal will increase the ability for that long-term potentiation to take place. So the more and more even weak signals you have, the better of an effect you're going to have, greater effect for long-term potentiation. So here's the receptors that are involved, right? So you have glutamate, that's going to be one of the main things that's going on here. Glutamate is one of the main neurotransmitters. NMDA receptors are going to be, they're considered coincidence detectors. Long story short here is that NMDA receptors are going to be blocked by magnesium, right? So it requires depolarization for that magnesium to be released, right? And once it does, then you have calcium especially coming into the cell. Again, depolarization, you're going to release magnesium from the NMDA receptor. Calcium is now going to enter into the cell, and that is what allows for long-term potentiation to take place. AMPA receptors are also going to be important for this process too, right? AMPA receptors, they also are going to be responding to glutamate. AMPA receptors, you're also going to see synaptogammin being all involved in this, right? Which, remember, that's going to be what allows for neurotransmitters to actually be released, right? So synaptogammin allows for the vesicles that contain the neurotransmitters to bind on to the cell membrane and then to ultimately allow for exocytosis of glutamate. So with AMPA receptor activation, you're going to be seeing strengthening of the response in the postsynaptic cell. So you're seeing stronger responses to glutamate. And then you're going to get upregulation of these types of receptors as well. Like the more you stimulate, the more receptors you're going to see, right? You're stimulating the excitatory postsynaptic potential in these cells. So here's a couple experiments that basically show what long-term potentiation looks like. You have just synaptic changes, dendritic changes within those cells. Yeah, this is FYI if you want to look into it. But long story short, just know that glutamate is going to be really important for this whole process. Then you have also things like you have the silent synapses. So stimulation of some glutaminergic synapses result in no firing of action potentials. But they fire when the postsynaptic cell is depolarized. So that's considered a silent synapse. So NMDA receptors are going to be involved in this. So this also changes over time. So when you're really, really, really young, you're only going to really see NMDA receptors. But into adulthood, you're going to see more and more AMPA receptors. More AMPA receptors means more stimulation, right? The NMDA receptor, which is blocked by magnesium, isn't going to be as easily activated. It needs to be totally depolarized for magnesium to get kicked out, right, to allow for calcium influx. Then when that happens, you're going to get more of these AMPA receptors coming into play. The AMPA receptors are what make these cells more sensitized to stimulation. And that's where you get long-term potentiation, long-term memory, the more of these AMPA receptors. That's why these increase over time into adulthood. So here's some more images of silent synapses. I'm not going to go into all the detail here with the different changes in microvolts, millivolts, and whatnot. Some more images of silent synapses. Again, I'm not going to go into too much detail about this, except for the fact that AMPA receptors increase more and more as you develop more memory. Calcium, of course, is going to be really important in this whole process, right? Calcium is going to be involved in long-term potentiation. You get calcium activating these transduction cascades, right? Because it acts as a second messenger. You also see your calcium calmodulin protein kinase. Remember CAM kinase. You also get activation of protein kinase C. So all these processes are going to allow for example, gene expression, right? So you get gene expression. You also get more transcription of these receptors, right? Transcription, translation of receptors. You get more AMPA receptor produced through transcription and translation. Here's these dendritic spines. Remember, those dendritic spines help to modulate activation of the cell. They can also be places where things can get stored, right? When you see dendritic spines. And then, again, glutamate release is going to be heavily involved in this whole process, right? When NMDA channels open up, after you get full depolarization of the cell, the calcium enters, so that's part of the whole cascade. Then you get AMPA receptors being activated. Then you get more and more AMPA receptors being placed into the phospholipid bilayer. And then you eventually get more sensitivity to glutamate. so this is the whole process. Increase glutamate sensitivity, increase in long-term potentiation. Late long-term potentiation. So in the long term, you're going to see more genetic expression, right? It's going to involve actual gene expression, so you can get proteins to be produced. You get remodeling of your synapses. If you subject a cell to a protein synthesis inhibitor, that's going to reduce this whole process, right? You're not going to get as much late-term, long-term potentiation. Changes in gene expression. So if you affect protein kinase, the protein kinase A, if you affect CREB, which is involved in transcription of genes, again, this is going to affect the formation of synapses. So if you inhibited protein synthesis, you're only going to see like the immediate effect of glutamate, but you're not going to see the long-term effect of glutamate, like long-term potentiation, which involves which receptor type? Is NMDA or AMPA? AMPA receptors. AMPA receptors, the more you're able to stimulate the NMDA receptor with a depolarization, the better you're going to be able to create more AMPA receptors. And the AMPA receptors are what really allow for long-term potentiation. Yeah, so it's a cascade of events, right? First event is you get depolarization with a strong stimulus. Second event is the NMDA receptor opens up because you release magnesium. Third event is you get more and more AMPA activation and production, allowing for more glutamate response. So in other words, if you block that process, if you block the ability for protein synthesis to take place, you're not going to see long-term potentiation. Again, gene regulation, gene transcription, right? You're getting more and more of these receptors being produced with long-term potentiation. So here's an example of a dendritic spine being formed as a consequence of long-term potentiation. You get more and more connections. So remember they were talking about those mossy fibers. Those mossy fibers, if you note, there's a bunch of dendrites with tons of the reason why they look mossy is because of all those dendritic spines. They're able to form lots of different connections, and that's important for the hippocampus for long-term memory. If you decrease the excitatory postsynaptic potentials, so you can see smaller rise in intracellular calcium, you're going to get loss of AMPA receptors. So you're not going to get as much AMPA receptor. You're not going to get in terms of like long-term memory formation. You're going to see a reduction in long-term memory formation. So the AMPA receptors are really, really important for long-term memory. What else here is important? So if you internalize those AMPA receptors, you're going to decrease the sensitivity to glutamate. So if you prune back AMPA receptors, which again, important for long-term memory formation, you're not going to be as sensitive to glutamate. So really important for long-term memory. Now we got a couple more minutes left. Let's talk about, well, let's close out with this. So you have Purkinje cells. So this is in the cerebellum. So we talked about mossy fibers and all that within like the hippocampus, but within the cerebellum, it's also going to be involved in, things like learning, also involved in, coordinating movements and things like that. So the cerebellum is really important for a lot of these processes as well. The type of fibers you find, you're going to see parallel as well as climbing fibers. And this is all involved in associated learning. So they're all going to be really close together, right? And so when the synapses fire, when they're close together, you're going to get associated learning with that whole process. And I'm not going to test you guys on all of these like second messenger system, but again, AMPA is going to be really important in this as well as glutamate. Glutamate is super important for this whole process to take place. I think that this is a good spot for us to close out for the day. We'll continue on here next time, and we'll finish out with seizures, different types of seizures and epilepsy. And then we're going to begin part 2 of this series, which is going to be involving like learning from experience. It's going to be like experience-based learning and neuroplasticity. Cool, guys. I'll see you all on Thursday.